Contrast learning ensures a strong separation between AI and human clusters in latent space. The projection head reduces overlap and improves interclass margins. Linguistic features introduce interpretability. For instance, human text tends to show more variation in sentence structure and lexical entropy. The lightweight neural classifier effectively combines all these signals while remaining efficient. So in summary, MISTI integrates contrastive representation learning with handcrafted linguistic and statistical features. It effectively detects AI-generated text by combining semantic understanding and surface-level cues. The model is lightweight, interpretable, and efficient for practical use. For the future, we plan to scale up testing to larger and more diverse datasets, extend MISTI to multilingual context, integrate rhetorical and discourse structures such as coherence.
Okay, Mr. CM Ansari,
Yes, sir.
you I see the data source is Kaggle.
Yes, sir. From Kaggle, I
What
have
is
used the asset.
the Kaggle? In Kaggle, we often see synthetically generated data also. Uh,
Sir, this
is there
dataset
any
has,
authentic? Is there
sir,
any? Who is the basically it's the basic contributor of the data?
sir, this dataset has already been used in already some published papers. So, and so that's why. So it was used henceforth. Since so in future work we want to work with that at
Okay, that was
interpretability.
my focus actually. Interpretability of the features is important. Okay,
Yes.
anybody has any question?
Sir, I want to know about the model. Actually, in your model, you use the Adam optimizer. So,
Yes.
how the Adam optimizer work in your model to obtain the optimal hyperparameter?
So for the neural network we use the so it was used in
So next presenter is paper ID number 145. Are you present here?
Yes, sir.
Uh, 145. Should we run it from here or she'll present it directly?
Sir, directly I present.
Oh, you want to present it directly? Okay. So please start.
Assalamu alaikum. Good afternoon, everyone. This is Khalilur Rahman. Today I will be presenting our research title on potato leaf net deep learning approach for potato leaf disease identification. This is our research outline: introduction, research gap and motivation, research objective, key contribution and novelties, dataset methodology, resultant discussion, and finally future work. Now start with the introduction. Potatoes are the third largest food crop worldwide and a key staple for over 1.3 billion people. But potato leaves are highly vulnerable to many diseases such as blight, fungal pest and viruses. This disease reduce both yield and quality. Understanding the background brings us to research gap and the motivation behind this study. Most of the previous studies are dependent on the pattern model like BGG or ResNet, GoogleNet, etc. Additionally, there is a lack of lightweight custom architecture specially designed for potato leaf disease detection. Since potato is a crucial crop, especially in Bangladesh, Bangladeshi farmers need a fast and reliable disease detection system. This gives motivated us to create a model. This research has to the following our objectives: to evaluate the CNN model and determine the best CNN model for detecting illness of potato leaves, to detect disease of the affected leaves, to create a new deep learning method for using custom CNN model named potato leaf net to classify potato leaf disease. This is our contribution. Developing custom CNN model, we propose a deep learning based custom CNN model named 42 LeafNet for leaf disease classification. Mm, Optimization method investigate and optimize various methods like Adam and stochastic gradient descent to fine tune models for better accuracy. And finally, we comparison and evaluation to different deep learning approach to determine the most effective strategies. This is our dataset section. This dataset contains 7 classes including early blight, late blight, fungal disease, plant disease, 42-6 nematoda, 42 virus, and healthy. Every classes has 400 training sample and 100 test sample and total 500. Before training, we applied processing technique to improve image quality and enhance model accuracy. This is our experimental setup. Our experiments were conducted using Python with TensorFlow and PyTorch. This setup gives us efficient train and evaluate all CNN model. This is our methodology part. This is architecture of our custom CNN model named 42 LeafNet. We applied 5 different models like GoogleNet, ShuffleNet, version 2, MNestNet, ResNet XT101, and finally our custom model 42 LeafNet. Now evaluation metrics. We used 4 metrics evaluation like accuracy, precision, recall, and F1 score. Here the model performance. GoogleNet achieved 82.4%, ShuffleNet version 2 92.6, MNestNet 84, ResNet XT101 93.4, and our custom model the highest accuracy 95%. This is our evaluation study of our custom model. We use different types of optimizer and different types of batch size and different used epochs. Finally, we used the best best hyperparameter and the Adam and best size 16 and the epochs 13 30 and the output the best accuracy 95%. To further analysis, this is our performance of proposed 42 LeafNet model, accuracy and loss. This is our accuracy graph and this is our loss graph. This is our ROC curve using 42 LeafNet. This is confusion matrix shows that nearly all prediction are correct across all classes. This is comparison to others model. Our 42 LeafNet model accuracy 95%, the highest than the others model. Despite high accuracy, some misclassification still occur. This is some mistake. Finally, our future direction, we expand the dataset with real field images, and we applied advanced data augmentation technique like GAN and others. And another future extension of this research could be implemented in a machine, mobile or web application for real-time quality inspection of. Thanks to all. I will be happy to answer any question.
Okay, thank you for your nice presentation.
Sir, I want to know about the model validation. You say your model provided a good result or best result, but is it a valid model?
Yes, sir. This is our
How can you say this is a valid model? Is there any cross validation? If you your model has no cross-validate result, then maybe it is a little bit confusing to say it is valid.
Because our architecture is a lightweight and custom built, because pretend model include a lot of layers, but our pretend our
Thank you.
So are there anyone? How many layers you used in your CNN, custom CNN? Could you please go to your slide? Model slide, yeah.
yes, sir. First of all, I used a ghost block and then conv 2D and then mesh normalization and then residual attention block and finally adaptive, then our output class.
So you showed the explainability of the model with your TNSA, right?
Yes, sir. TSN graph we are checked our dataset.
Okay, thank you. Thank you for your presentation. We may move to next one.
Thank you, sir.
Paper ID 165. Are you present here? If you are present, then we can run the slide for you. We can
Oh, yeah.
run your presentation.
Sure.
Will you
I'm going to
present
the
it directly or should we run your video recording?
Oh, you can share your screen. I mean, the presentation slide. It's okay.
Okay.
Very pleased to present our current research. Hello everyone. My name is Deeptapal, representing the American International University of Bangladesh, and my co-author is Ikramul
Sir,
Hassan. He's from
Can you please share? I mean, the presentation slide. Then I can.
it is very pleased to present our current research title analyzing genetic diabetes trends in the USA using data mining driven prediction to identify how family history, gender, and age affect likelihood the insulin dependency. The ultimate goal was to develop a reliable and interpretable predictive model that could support early diagnosis and personalized healthcare planning. The main objectives of our study were three: collected data from 200 diabetes affected individuals equally divided between the male and female participants. The dataset included demographic details, clinical informations, and mostly importantly, the diabetic history of the both parents and grandparents. The data were pre-processed in Microsoft Excel and analyzed using the Weka software. The three core algorithms were applied, simple k-means for the clustering similar patients groups, J48 decision tree for classifications, and simple linear regressions for predictive analysis. These combinations enabled both interpretability and quantitative accuracy in identifying the genetically influenced diabetes patterns. The results from clustering revealed four distinct patient groups. When both parents and grandparents were diabetic, the cases were strongly genetic, accounting from 44 instances. In contrast, 96 cases showed no genetic connections when neither ancestors had diabetes. Intermediate cases were also observed, 47 when the parents were diabetic and 13 when only the grandparents were affected. The decision tree generated from the data provided a clear and intuitive rule. If any ancestors has diabetes, the case is likely genetic. This interpreticity makes the model highly suitable for clinical applications. Gender-based predictions further reinforce the findings. Among 100 male and 100 female participants, the predicted number of genetically inherited cases closely matched actual records, differing by only 2 instances for each gender. The study also analyzed insulin dependency patterns and found a nearly balanced rate, around 46 to 48% across both genders. The 21 to 40 years age group demonstrated the highest insulin dependency, indicating that genetic diabetes may manifest most strongly during early adulthood. While the outcomes were encouraging, we also recognize the study's limitations. The dataset was relatively small and limited diversity, which may constrain the model's generalization. Furthermore, factors such as lifestyle, diet, and environmental conditions were not incorporated into the analysis. In future work, we aim to include larger and more diverse datasets, integrate genomic and biomarker information, and possibly connect the predictive. In conclusion, this research demonstrates how data mining techniques can effectively identify genetic diabetes pattern and provide interpretable predictive insights. By combining clustering, decision tree, and regression methods, the framework successfully highlights the influence of the family history. Although preliminary, the results emphasize the potential of data-driven healthcare analysis to support early detection and personalized management of diabetes. With future enhancement, this model could contribute meaningfully to clinical decision support and public health planning. That's all about our presentation, and thank you very much.
ওই চলে গেল। We cannot see you here. Mr. Diptopal, question and answer session. You have to attend it.
Sir, I think maybe Deepto is not available, so you can ask any if you have any question, then ask me. No problem. I will answer the question.
Okay, on behalf of him, Mr.
Yeah.
Ikramul